Hello there, and welcome to Sleepy Science Vault. Pain is one of the most universal experiences a human being can have. Every person alive has felt it, and most of us assume we understand it. But what if almost everything we believe about pain is wrong? What if pain isn't something that happens to you, but something your brain actively creates? That idea might feel strange at first. But by the time you reach the end of this, the way you think about pain will have quietly, fundamentally shifted. Let's begin at the beginning. For most of human history, pain was understood as a simple alarm system. You touch something hot, you feel burning. You cut your hand, you feel the cut. The logic seemed obvious. Damage happens in the body, pain travels up to the brain, and you feel it. Clean, direct, mechanical. But that's not how it works. Not even close. Here is one of the most important things modern neuroscience has discovered about the human body. Pain is not a signal that travels from your tissues to your brain. Pain is something your brain produces. It is an output, something the brain generates, not an input it passively receives. This changes everything. Your brain is constantly receiving information from millions of sensory nerves spread throughout your body. Those nerves are monitoring temperature, pressure, chemical changes, tissue stress. They are sending data upward, continuously, every second of your life. But that data is not pain. It is simply information. What happens next is far more interesting. Your brain takes all of that incoming data, weighs it against everything it knows, your memories, your current situation, how safe you feel, what is at stake, and then it makes a decision. And only if your brain decides that the body needs protecting, that something requires urgent attention, only then does it generate the experience of pain. Pain is a decision. Not a conscious one. Not something you choose in any deliberate way. But a decision nonetheless, made by the most complex structure in the known universe, acting on your behalf, trying to keep you alive. Now, the cells responsible for starting this whole process have a name: nociceptors. These are specialized sensory nerve endings, woven into nearly every tissue in your body. Skin, muscle, joints, organs. They are not pain receptors. That distinction matters. They don't detect pain. They detect potential danger, signals of damage, or the threat of damage. Some nociceptors respond to heat, specifically to temperatures that rise high enough to risk burning the tissue. The threshold is around 43 degrees Celsius. Below that, warmth feels pleasant, even comforting. Above it, those receptors begin to fire, sending urgent messages upward toward the brain, saying, "This is getting dangerous." Others respond to mechanical pressure, the kind of force that might crush or tear. Others are chemically sensitive, tuned to specific molecules that appear in the body when something goes wrong. When you sprain an ankle or bruise a muscle, the damaged cells release a cascade of inflammatory chemicals. Prostaglandins, bradykinin, histamine. These substances flood the surrounding tissue, and nociceptors that would normally stay quiet suddenly become exquisitely sensitive. The area swells. It becomes warm. And even light touch that would otherwise mean nothing becomes intensely uncomfortable. This is inflammation doing exactly what it was designed to do, drawing the brain's attention to a region that needs care. Lactic acid is another trigger, and one that many people have felt without realizing what it was. During intense physical effort, when your muscles are working faster than your body can deliver oxygen, lactic acid accumulates in the tissue. Nociceptors respond to that acidic environment directly. The deep, burning ache of an exhausted muscle, that specific feeling, is partly lactic acid speaking to your nervous system in its own chemical language. But here's where things become genuinely remarkable. All of this, the nociceptors firing, the inflammatory signals, the chemical messages traveling up through the spinal cord, none of it guarantees that you will feel pain. Because the brain is not a passive receiver. It is an interpreter, an editor. And sometimes, depending on context, it chooses to suppress the signal entirely. Soldiers wounded in combat have reported feeling nothing at the moment of injury. Athletes have finished races on broken bones. People in accidents have calmly walked away from damage that should, by any mechanical logic, have been agonizing. The injury was real. The nociceptors were almost certainly firing. But the brain decided something else was more important. And on the other side of that same coin, people can feel profound, disabling pain without any detectable injury at all. Scans show nothing. Tests reveal nothing. The tissue is intact. And yet the pain is absolutely real. It is not imagined. It is not weakness. It is the brain generating a genuine pain experience in the absence of physical damage, because it has concluded, for reasons of its own, that the body is under threat. Injury does not equal pain. Pain does not equal injury. The two are related, but they are not the same thing. The level of pain you feel does not correspond reliably to the size of a wound or the severity of tissue damage. A paper cut across a fingertip can feel disproportionately sharp. A deep internal injury can sometimes go nearly unfelt. What determines how much pain you experience is not the objective state of your body. It is your brain's assessment of how much danger you are in, and how urgently it wants you to change your behavior. Pain is protection. It is your brain's most persuasive way of saying, pay attention to this. Stop. Rest. Change course. It is not punishment. It is not damage itself. It is a message, constructed, tailored, and delivered by a brain that is, at its core, trying to keep you whole. And that is only the beginning of the story. Let's take a step back and think about something ordinary. You are moving through your day, and without realizing it, your body is quietly receiving thousands of signals every single moment. Pressure from the chair beneath you. The temperature of the air on your skin. The faint stretch of muscle as you shift your weight. All of that information is flowing inward, continuously, and almost none of it reaches your conscious awareness. Your nervous system is extraordinarily good at filtering. Most of what it receives, it quietly discards. And this is not a flaw, it is an elegant design. If you were conscious of every signal your body sent, you would be overwhelmed within seconds. But some signals do get through. And the reason they get through is something called a threshold. pain does not simply switch on the moment a nociceptor fires. There is a level, a line that must be crossed, before the brain begins to take a signal seriously. Below that threshold, a stimulus might register as warmth, or pressure, or mild discomfort. Something you notice and then set aside. But once a signal is strong enough, persistent enough, or arrives in sufficient numbers, it crosses into territory the brain can no longer comfortably ignore. That threshold is not fixed. It shifts. It changes depending on your body's state, your emotional condition, how rested you are, whether you feel safe. People who are anxious, exhausted, or under sustained stress often find their threshold lowers. Things that would ordinarily feel manageable begin to feel sharper, more intrusive. Conversely, in moments of deep focus or calm, the threshold can rise. The same stimulus that would have been bothersome in a tense moment passes almost unnoticed in a peaceful one. This is why pain is so personal. Two people can experience the same physical event and walk away with entirely different impressions of how much it hurt. Neither of them is exaggerating or minimizing. Their thresholds were simply different. Their brains were working from different starting conditions. Once that threshold is crossed, however, a journey begins. And it is a journey worth understanding, because it is happening inside you, right now, in this very moment, in quiet ways you have never seen. A nociceptor fires somewhere in the body. Maybe in a fingertip, maybe deep in a muscle, maybe in the lining of a joint. That electrical signal does not teleport to the brain. It must travel. And to travel, it moves along a nerve fiber, a thin biological wire, stretching from wherever the signal originated, upward through the body, toward the spinal cord. These nerve fibers are not all the same. This is one of the details that tends to surprise people when they first hear it, and it explains something that most of us have experienced but never had the words for. There are two main types of pain fibers, and they move at very different speeds. The first type is called A-delta fibers. They are insulated, wrapped in a fatty sheath called myelin that allows signals to travel quickly. When an A-delta fiber carries a pain signal, it arrives fast. The pain it produces is sharp, clear, immediate. It has a precise location. It is the first thing you feel when you stub your toe or touch something unexpectedly hot, that sudden, bright, exact sensation that makes you pull away. The second type is called C fibers. These are much slower. They carry unmyelinated signals, bare, uninsulated, and the information they transmit arrives later. Several seconds after that first sharp flash. The pain they produce is different in character, dull, deep, spreading, aching. It is the throb that follows the initial sting. The soreness that lingers. The heavy, diffuse discomfort that makes you want to stop using a limb and simply let it rest. Two waves, from the same event, arriving in sequence. First the sharp warning. Then the slow, insistent ache. You have felt this your entire life without knowing there were two separate conversations happening, two different types of fibers delivering two different kinds of messages, on two slightly different timelines. The nerve fibers carrying these signals converge at the spinal cord. This is a critical way station in the journey, and far more than just a passive relay. The spinal cord does not simply pass signals upward unchanged. It processes them. It weighs them. And crucially, it can amplify them or dampen them depending on the signals it receives from the brain above. Here the signal crosses what is called a synapse, a gap between nerve cells. To cross that gap, the electrical signal must be converted into a chemical one. The nerve releases molecules called neurotransmitters, which drift across the microscopic space and bind to receptors on the next cell. That binding triggers a new electrical signal, and the message continues upward. This process, electrical to chemical to electrical again, happens countless times across the nervous system. It is the language of your entire inner world. One of the most important neurotransmitters in this process is called substance P. It is released at the spinal cord level and plays a significant role in amplifying pain signals, essentially turning up the volume on the message before it continues its journey toward the brain. Other neurotransmitters, like enkephalins and endorphins produced by the body's own internal systems, can do the opposite, quieting the signal, softening it, easing its passage. From the spinal cord, the signal ascends to the brain. And here, most of the body follows a fairly consistent path. But the face is different. Pain signals from the face, from the teeth, the sinuses, the skin of the cheek, the forehead, the jaw, do not travel the same route. They travel along a separate cranial nerve called the trigeminal nerve, one of the largest sensory nerves in the body, which feeds directly into the brainstem without following the spinal pathway at all. This is why dental pain, headaches, and facial injuries have such a distinctive, often overwhelming quality. The trigeminal system is ancient and sensitive, and the regions of the brain it connects to process facial signals with particular intensity. Once a pain signal reaches the brain, it does not land in a single location. There is no one place in your brain where pain simply lives. Instead, the signal distributes across multiple regions simultaneously, and two broad pathways handle different aspects of what you ultimately experience. The first pathway is sometimes called the sensory discriminative pathway. This is the part of the experience that tells you where the pain is, what kind of pain it is, and how intense it seems. It is the factual, informational dimension, processed largely through the somatosensory cortex, which contains a detailed map of the entire body surface. The second pathway is the affective motivational pathway. This is the part that makes pain feel unpleasant. This is where the suffering lives, the emotional weight, the urgency, the need to make it stop. This pathway involves areas like the anterior cingulate cortex and the insula, regions deeply connected to emotion, memory, and the sense of self. Remarkably, studies have found that when people are given drugs that block this pathway, they can still feel pain in a sensory sense. They know it is there, they can locate it, but they report that it no longer bothers them. The sensation is present. The distress is gone. This tells us something profound. Pain is not one thing. It is at least two things, sensation and suffering, bound together but ultimately separable. And your brain is managing both, simultaneously, every time pain arises. Now, before any of this reaches consciousness, before you are even aware that something has happened, your body has already begun to respond. The spinal reflex is faster than thought. When you touch a hot surface, your hand withdraws before your brain has registered what occurred. The signal from your fingertips travels to the spinal cord, and at the spinal cord level, without waiting for permission from above, a motor command is issued: pull back. The hand moves. Only a fraction of a second later does the pain signal complete its journey to the cortex and become conscious experience. You felt yourself pull away before you felt the burn. This reflex is extraordinarily ancient. It exists in virtually all vertebrates. It is one of the body's deepest, most fundamental protections, an automatic, local circuit that acts before the higher brain can deliberate. At nearly the same moment, a broader alarm is triggered throughout the body. The stress response, driven by the sympathetic nervous system, activates almost instantly. Adrenaline releases into the bloodstream. Heart rate increases. Breathing quickens. Blood is redirected away from the digestive system and toward the large muscles. Pupils dilate. The body is preparing, within milliseconds, to fight or to flee. This is your body in its most ancient mode, mobilizing every available resource, sharpening every sense, narrowing your attention to the immediate threat. It is not a malfunction. It is a masterpiece of biological engineering, refined across hundreds of millions of years of evolutionary pressure. Every component of that response was shaped by survival. And yet, in the modern world, where most of the pain we experience is not a predator or a cliff edge but a headache, or a sore back, or chronic discomfort with no clear exit, that same ancient system can become a source of exhaustion. The body keeps preparing for a threat it cannot outrun. The alarm keeps sounding when there is nothing to escape. Understanding this, really sitting with it, changes how you might relate to your own pain. Not to dismiss it, never to dismiss it, but to recognize that behind every ache, every throb, every sharp moment of discomfort, there is a biological system of breathtaking sophistication, doing exactly what it was built to do. It is trying, in the only language it knows, to protect you. By the time a pain signal reaches the brain, it has already traveled an extraordinary distance. It has crossed from the outer edges of the body inward, jumped synapses in the spinal cord, converted from electrical impulse to chemical messenger and back again, and ascended through the brainstem toward the higher structures of the brain. And now, at the threshold of conscious experience, it arrives at a small but critically important structure buried deep in the center of the brain. The thalamus. Think of the thalamus as the brain's central relay station, a dense, walnut-sized cluster of neurons that sits at the crossroads of almost all incoming sensory information. Nearly everything your brain receives from the outside world passes through here first. Vision, hearing, touch, temperature, and yes, pain signals from the body. The thalamus receives them, sorts them, and routes them onward to the appropriate regions of the cortex above. But the thalamus is not simply a passive switchboard. It is an active gatekeeper. It can enhance signals or suppress them. It can hold information briefly or dispatch it immediately. And in the case of pain, its role is particularly significant, because the paths it chooses to activate next will determine not just whether you feel pain, but what kind of pain you feel, where you feel it, and how deeply it affects you. From the thalamus, signals fan outward across multiple brain regions simultaneously. And this is one of the most important things to understand about how pain actually works inside the brain. There is no single location where pain happens. There is no one spot you could point to on a diagram and say, there, that is where pain lives. Pain is distributed. It is a whole brain event. The somatosensory cortex receives the signal and begins to map it onto the body. This region of the brain contains what neuroscientists sometimes call a homunculus, a kind of internal body map, or representation of the entire physical self laid out across the surface of the cortex. Different areas of this map correspond to different parts of the body: the hands, the face, the feet, the torso. When a pain signal arrives, the somatosensory cortex locates it on this map and begins to answer the practical questions. Where is this coming from? How intense is it? Is it sharp or dull? Burning or aching? Localized or spreading? This is the brain doing its investigative work, assembling the factual picture of what is happening and where. At the same time, the insula is activating. This is a region folded deep within the brain, hidden from the outer surface, and it plays a remarkable role in translating raw sensation into felt experience. The insula connects the physical with the emotional. It is involved in body awareness, in the sense of what your body feels like from the inside, and in the integration of sensation with meaning. When the insula engages with a pain signal, it begins to give that signal emotional texture. This isn't just data anymore. This is something that matters to you. Simultaneously, the anterior cingulate cortex comes online. This region, as we touched on earlier, handles the suffering dimension of pain, the part that makes it feel urgent, distressing, impossible to ignore. Research involving patients with certain types of brain lesions has shown that damage to this area can produce a strange condition where a person acknowledges feeling pain but no longer finds it troubling. They know it is there. They can describe it. But the emotional charge, the need to make it stop, has been removed. The sensation without the distress. The prefrontal cortex, the brain's seat of reasoning and decision-making, also activates. It is trying to make sense of what is happening, to contextualize the signal, to decide what to do. Do we move? Do we rest? Is this familiar? Is this serious? The prefrontal cortex brings memory and judgment into the conversation. And then there is the amygdala, the brain's emotional alarm center, deeply involved in fear and threat detection. When pain signals arrive, the amygdala is paying close attention. It is asking a question that is older than language: am I in danger? Its response shapes the emotional coloring of the whole experience. All of these regions, the somatosensory cortex, the insula, the anterior cingulate, the prefrontal cortex, the amygdala, and several others, activate together, weaving their individual contributions into a single, unified experience. What you feel as pain is not one signal arriving in one place. It is a conversation happening across an entire network, all at once, in parallel. The technical term neuroscientists use for this collective pattern of activation is the pain neuromatrix, a distributed network of regions that together construct the experience. And yet, even with all of this activity happening, even with signals firing across an entire network of brain structures, pain still does not become conscious until one final condition is met: attention. This is one of the quieter revelations about how pain works, and it is one that carries enormous practical weight. The brain must allocate attention to a signal before it rises to the level of conscious experience. Signals can be traveling, being processed, activating regions, and if the brain's attentional resources are directed elsewhere, they may not become felt pain. This is why people engaged in moments of intense focus, athletes in competition, surgeons in the middle of delicate procedures, people lost in creative work, can sustain injuries without feeling them until later. The signal was there. The tissue was damaged. The nociceptors were firing. But the brain's attentional spotlight was pointed somewhere else entirely. And without that spotlight, pain remained below the surface of awareness. The moment focus shifts, the moment the race ends, the surgery concludes, the flow state breaks, the signal is suddenly, sometimes startlingly, felt. The brain looks at what has been quietly waiting for it, and the pain arrives all at once. This is not the body holding out on you. It is attention doing its job, managing the finite resource of conscious awareness with extraordinary pragmatism. Now, once pain does reach consciousness, something fascinating begins to happen. Because the brain does not experience raw data. It interprets. And the interpretation of pain is one of the most context-dependent processes in all of human biology. The same physical signal, the same input to the same type of nerve fiber, can feel profoundly different depending on the circumstances surrounding it. The brain is constantly asking not just what is this signal, but what does this signal mean right now, in this situation, for this body. Consider something as simple as how pain feels different at different times of day. The same aching back that is barely noticeable in the morning might feel significantly more intense late at night, when there are fewer distractions, when the body is still, and when the mind has nothing else to attend to. The tissue has not changed. The signal has not grown. But the brain's context has shifted, and so the interpretation shifts with it. Or consider the role of meaning. There is a classic study in pain research involving soldiers wounded in combat who required far less pain medication than civilians with injuries of comparable severity. The researchers eventually understood why. For the soldiers, the wound carried a meaning beyond the injury itself. It meant survival, it meant leaving the battlefield, it meant going home. The context transformed the signal. The brain, assessing the situation, determined that aggressive pain amplification was not warranted. The meaning of the injury changed the experience of it. This contextual layer of interpretation also produces the extraordinary diversity of sensations that fall under the single word pain. Because pain is not one feeling. It is a whole family of experiences, each with its own character. There is the bright, clean sharpness of a fresh cut. The deep, nauseating throb of a bruised bone. The hot, spreading burn of inflamed tissue. The tight, gripping squeeze of a muscle in spasm. The dull, relentless ache that settles in and refuses to leave. the strange, electric shooting sensation of a nerve under pressure. The hollow, interior ache that seems to have no precise location at all. Each of these is a different message, constructed by the brain using different combinations of signal types, different patterns of neural activation, different weightings from the emotional and sensory networks. The brain is not generating one experience and calling it pain. It is generating many experiences, each tailored to communicate something specific about the nature of the threat. And then there is the dimension of individual difference, the way pain is not experienced equally across people, and cannot be. This is something that medicine has been slow to fully appreciate, and it has caused real harm over the years. For a long time, there was a tendency to assume that pain was objective, that a particular injury or condition should produce a predictable amount of pain in a predictable range of people. If someone reported more pain than expected, it was tempting to question them. To assume they were being dramatic, or overly sensitive, or somehow not coping well. But the science is unequivocal. Pain experience is genuinely, deeply, profoundly individual. It varies based on genetics. Some people carry gene variants that make their nociceptive system more or less sensitive at a biological level, entirely independent of psychology or upbringing. It varies based on prior experience. A person who has lived through significant pain in the past has a nervous system that has been shaped by that experience, and that shaping is real and physiological. It varies based on culture, which influences how pain is expressed, how it is interpreted, how much attention is given to it. It varies based on the current state of the nervous system, how much inflammation is present, how well someone has slept, how chronically stressed they are. Pain tolerance is not a measure of strength. It is not a measure of courage or resilience or character. It is a measure of a specific biological system operating under specific conditions in a specific body. Comparing one person's pain threshold to another's is like comparing their height and calling the taller person more disciplined. The comparison simply does not mean what people assume it means. Two people can sit side by side, experience an identical stimulus, and come away with genuinely different experiences of how much it hurt. Both are telling the truth. Both are reporting their reality accurately. Their nervous systems are simply different, shaped by different genetics, different histories, different chemistry, different lives. What this means, at its heart, is that pain is always personal. There is no objective measure waiting somewhere that could validate or invalidate what any individual person feels. The experience is real because it is being generated by a real brain, in a real body, responding to the world as that body has learned to respond to it. When someone tells you they are in pain, they are not offering an interpretation. They are reporting a fact about their own inner world, a world that only they can directly access, and that deserves nothing less than to be believed. There is something quietly extraordinary about all of this, if you let yourself sit with it. The experience you have known your entire life as pain, something that can feel so raw, so immediate, so physical, is at the same time one of the most sophisticated acts of interpretation the universe has ever produced. Billions of neurons, coordinating across dozens of regions, drawing on memory and emotion and context and genetics, to construct something that feels like the simplest, most undeniable thing in the world. something that hurts. And in that construction, in that extraordinary biological effort, there is a kind of care, an ancient, impersonal, tireless care, for the fragile and remarkable thing that is a living human body. There is a question that pain researchers have been asking for decades, and it is one that touches something deeply personal for almost everyone who hears it. Why do some people seem to move through physical hardship with relative ease, while others find the same experiences genuinely, significantly harder to bear? Why does the same injury, the same procedure, the same chronic condition produce such different outcomes in different people, even when every other variable seems equal? For a long time, the answer was assumed to be psychological. Attitude, willpower, mental toughness. The belief was that if someone struggled more with pain, the difference must lie somewhere in their character, their approach, their emotional resilience. But science has been quietly dismantling that assumption for years. And what it has found in its place is something far more interesting, and far more humbling. Some of the difference begins before you are even born. Genetics play a genuine, measurable role in how a person experiences pain. This is not a metaphor. It is not a generalization. It is a finding supported by decades of research, twin studies, and increasingly detailed genomic analysis. Your DNA contains instructions for building the proteins that form your nociceptors, your nerve fibers, your neurotransmitter receptors, your brain's own pain-modulating systems. Small variations in those instructions, variants that are common in the population, that you inherited without any choice in the matter, can make your nociceptive system more or less reactive at a fundamental biological level. Some people are born with nociceptors that have a lower activation threshold. Their pain-detecting system is, from the outset, more easily triggered. This is not a defect. In certain environments across human history, it may have been a survival advantage, heightened sensitivity to threat, a faster warning system, a body that responded more urgently to potential damage. But in the context of modern life, where chronic pain is one of the most widespread sources of suffering on the planet, it is a biological reality that deserves acknowledgement rather than judgment. There are also genetic variants that affect how efficiently the brain's own pain-dampening systems function. The brain produces natural opioid-like molecules, endorphins, enkephalins, that can reduce the intensity of pain signals at multiple points along their journey. Some people, by genetic inheritance, produce these molecules more effectively, or have receptors that respond to them more strongly. Others have systems that are less efficient. The difference in pain experience between these two people, encountering the same stimulus, can be substantial, and neither of them chose the system they were given. Pain sensitivity, in this sense, can be genuinely inherited. It can run in families not because of shared attitudes or learned behaviors, though those are real too, but because of shared biology, shared proteins, shared receptor structures, shared neurochemistry passed down through generations. Knowing this does not solve pain. But it does something important. It moves the conversation out of the realm of judgment and into the realm of understanding. And that shift, quiet as it sounds, can be transformative for someone who has spent years being told, in one way or another, that their pain is somehow their fault. but genetics is only one thread in a far more complex story. Because the experience of pain does not live in biology alone. It lives at the intersection of biology and everything else: the mind, the emotions, the relationships, the environment, the history a person carries inside them. And this intersection is where things become genuinely fascinating. The connection between psychological state and pain experience is not soft science. It is not a polite acknowledgement that stress can make things feel worse. It is a hard, neurological, measurable reality. The brain structures that process pain and the brain structures that process emotion are not separate systems that occasionally influence each other. They are deeply, anatomically intertwined. The anterior cingulate cortex, which contributes so significantly to the suffering dimension of pain, is also central to emotional processing. The insula, which gives pain its felt, interior quality, is also heavily involved in anxiety and interoception, the sense of what is happening inside the body. The amygdala, the brain's threat detection center, is in constant communication with the pain processing network. These systems share anatomy. They share chemistry. They are, in a very real sense, the same system wearing different hats. Anxiety and stress do not merely accompany pain. They actively amplify it. Where a person is anxious, the amygdala is in a heightened state of alert, scanning for threat, primed to react. In this state, the pain processing network is more easily activated, and signals that might have been filtered or dampened in a calm state are instead amplified and allowed through. The threshold drops. The gain turns up. Pain that might have been manageable in a restful, safe moment becomes something significantly harder to bear. Stress hormones, particularly cortisol, have direct effects on the nervous system that increase nociceptive sensitivity over time. Chronic stress means chronic cortisol exposure, which gradually sensitizes the pain pathways, making the system progressively more reactive. A person who has been under sustained stress for months or years is not imagining that they hurt more easily. Their nervous system has been chemically altered by that stress in ways that are entirely real and measurable. Emotions, more broadly, change the intensity of pain experience in both directions. Fear makes pain worse. Sadness tends to make pain worse. Anger can, in some contexts, temporarily suppress pain. There is an evolutionary logic to this, since pursuing or escaping a threat requires the ability to ignore immediate discomfort. Joy, connection, laughter, and feelings of safety tend to reduce pain. Not metaphorically. Neurochemically. The release of oxytocin, associated with feelings of love and social bonding, has measurable pain-reducing effects. The experience of genuine pleasure activates dopaminergic pathways that interact directly with the pain-modulating system. Your emotional state is not a footnote to your pain experience. It is one of its primary authors. The environment in which pain is experienced matters too, in ways that are immediate and profound. Experiments have shown that people rate the same painful stimulus as more intense in cold, harsh, clinical environments compared to warm, comfortable, aesthetically pleasant ones. The presence of natural light, soft colors, familiar objects, or pleasant sounds has been shown in research settings to measurably reduce pain scores. hospitals that have incorporated natural elements, windows with views, access to gardens, gentle acoustics, have found statistically significant differences in patient recovery and pain medication use. This is not about distraction, exactly. It is about what the environment signals to the brain. A cold, sterile, uncomfortable environment is a place that feels unsafe. And an unsafe environment is a place where the brain's threat detection system remains activated. Where pain thresholds stay low. Where the amygdala keeps its watch. A warm, comfortable, familiar environment signals something different. It signals that the immediate world is not dangerous. And in that context, the brain can ease its vigilance. The pain-modulating system can function more effectively. The whole system breathes a little. This is why the brain, in certain situations, will generate pain that seems to have no physical origin. When a person is in a situation that feels genuinely unsafe, whether physically, emotionally, or socially, the brain sometimes responds by producing pain as a kind of alert signal. Not because tissue is damaged. Not because a nociceptor has fired. But because the brain has assessed the current situation and concluded that the body needs to withdraw, to be protected, to be removed from where it is. The pain is real. The danger that prompted it may be social or emotional rather than physical. But the brain does not always distinguish cleanly between these categories. Threat is threat. Danger is danger. And the response is the same system, activated by the same alarm. Trauma occupies its own profound place in this story. When a person has lived through significant trauma, particularly early in life, when the nervous system is still forming, the effect on the pain processing system can be lasting and deep. Trauma does not just leave psychological marks. It leaves neurological ones. It alters the structure and sensitivity of the stress response system. It changes the resting state of the amygdala. It shifts the baseline of the entire threat detection apparatus toward higher vigilance, toward a nervous system that has learned, through painful experience, that danger can arrive without warning and that it is better to stay alert than to relax. For people living with the legacy of trauma, pain is often amplified, not because they are fragile, not because they are not trying, but because their nervous system is running a different program. A program that was written under extraordinary duress, that served a real protective purpose at the time it was created, and that now continues to run even when the original threat is long past. Understanding this changes everything about how we should approach pain in people who have been through difficult things. The question is never why are they so sensitive. The question is always what has their nervous system learned, and what does it need to feel safe. And then there is memory. Because pain is not only shaped by what is happening now, it is shaped by what has happened before. The brain is a prediction machine. At every moment, it is drawing on past experience to anticipate what the present moment means and what the future is likely to hold. This is not a quirk of human psychology, it is the fundamental operating principle of the brain. Expectation is built into every perception, including pain. When you have experienced pain in a particular context before, the brain encodes that information. It remembers. Not in the way you consciously remember a childhood birthday, but at a deeper, more automatic level, the way your body remembers how to ride a bicycle or how to read words without sounding out each letter. The memory is stored in the pattern of neural connections, in the sensitized pathways, in the tuned and calibrated networks of the pain processing system. The next time that context arises, the same type of movement, the same environment, the same situation that was previously associated with pain, the brain does not wait to see what happens. It begins preparing. It anticipates. And in anticipating pain, it can begin to generate the neurological conditions for pain before any nociceptor has fired a single signal. This is why a person who has hurt their back before feels apprehension, and sometimes pain, simply when they approach the movement that caused the original injury. The tissue may be fully healed. The original source of damage may be completely resolved. But the brain has a memory, and that memory is exerting real influence over what is felt in the present. It is also why expectations about pain, formed from prior experience, from things people have been told, from beliefs about what a particular condition or procedure will feel like, have such a significant effect on what is actually experienced. Patients who are told a procedure will be painful consistently report higher pain levels than patients given the same procedure with neutral or reassuring framing. Not because they are imagining it. Because expectation primes the brain's pain system, and a primed system is a more responsive one. This phenomenon has a name, nocebo. It is the dark mirror of the placebo effect. Where placebo is the brain generating positive outcomes in response to the expectation of benefit, nocebo is the brain generating negative outcomes, including pain, in response to the expectation of harm. Both are real. Both involve genuine neurochemical changes. Both demonstrate, again and again, the extraordinary degree to which the brain's predictions shape the body's experience. Pain, in this sense, is always partly a story the brain is telling, a story informed by everything that came before, everything that surrounds the present moment, everything that feels true about the world and the body and what they are capable of. The biology is real. The signals are real. But the story is built for more than signals. It is built from a life. And that makes the experience of pain one of the most deeply personal things a human being can carry, something that cannot be fully seen from the outside, cannot be accurately judged, and cannot be understood without first understanding the person inside whom it lives. Something changes when pain stays too long. Not just emotionally, not just psychologically, though it changes those things too, but structurally, chemically, at the level of the neurons themselves. The nervous system is not a fixed system. It is a living one, and like all living things, it adapts to its conditions. When pain signals pass through the nervous system repeatedly, over days and weeks and months, the pathways those signals travel begin to change. The neurons involved become more efficient at transmitting pain. Their thresholds lower. Their sensitivity increases. They begin to respond to stimuli they would previously have ignored entirely, and to amplify stimuli that would previously have felt mild. This process has a name, central sensitization. And it represents one of the most important, and most challenging, aspects of how chronic pain works. Think of it this way. When you walk the same path through tall grass again and again, the grass flattens. The path becomes clearer, easier to travel, more defined with each crossing. The nervous system works similarly. Repeated pain signals carve their paths deeper into the neural architecture. The route becomes easier to activate. Less input is required to produce the same output, or more. In the short term, this sensitization serves a purpose. After an injury, the affected area becomes more sensitive as a way of protecting it during healing. Touch that would normally be neutral becomes uncomfortable. Movement that would ordinarily be painless becomes cautious and deliberate. The body is enforcing rest, enforcing care, enforcing attention to a region that needs to recover. But when the original injury heals and the sensitivity remains, when the pathway stays carved deep even after the reason for carving it has passed, sensitization stops being protective and becomes a problem of its own. The alarm system that was built for a specific threat continues sounding long after the threat has resolved. And the longer it sounds, the more sensitive it becomes to its own input, and the louder it grows. This is part of what makes chronic pain so genuinely different from acute pain. It is not simply pain that has lasted longer. It is pain that has, in a meaningful sense, changed the system that generates it. It is pain that has left marks. And yet, and this is where the story begins to shift in a more hopeful direction, the same plasticity that allows the nervous system to become more sensitive to pain also allows it to move in the other direction. The same capacity for change that creates the problem also holds the possibility of relief. Because the brain is not only capable of amplifying pain, it is equally capable of reducing it, suppressing it, overriding it entirely. And it does this constantly, through a range of mechanisms so sophisticated that they continue to surprise researchers who have spent careers studying them. The brain modulates pain. It turns the signal up when circumstances require heightened sensitivity. And it turns the signal down when circumstances allow, or when something more important demands the body's full resources. The theoretical framework that first gave scientists a clear way to understand this bidirectional control came from a landmark paper published in 1965, written by two researchers named Ronald Melzack and Patrick Wall. They called it the gate control theory of pain. And it genuinely changed everything. Before gate control theory, pain was understood as a one-way system. Signals traveled from the body to the brain, and the brain felt them. That was the model. Simple, linear, mechanical. Melzack and Wall proposed something different. They described a gating mechanism in the spinal cord, a biological switch, located at the point where sensory nerve signals first enter the central nervous system, that could open or close based on input from multiple directions. Not just from the nerves below, but also from the brain above. When the gate is open, pain signals pass through freely and ascend to the brain where they become experience. When the gate is closed, or partially closed, those signals are reduced or blocked before they ever reach conscious awareness. And crucially, the brain itself can send signals downward through the spinal cord to influence the gate's position. This was revolutionary. It meant the brain was not just a passive recipient of pain. It was an active participant in regulating it. It could reach down into the spinal cord and, through descending neural pathways, either amplify or dampen incoming signals before they were allowed to complete their journey. Gate control theory has been refined significantly in the decades since 1965. Some of the specific cellular mechanisms Melzack and Wall originally proposed have been updated as neuroscience has advanced. But the central insight, that pain is actively modulated, that there is a bidirectional control system, that the brain can both generate and suppress pain experience, has proven not just correct but foundational. It is the basis for understanding almost everything that followed. One of the most important ways the brain reduces pain is through its own internal chemistry. Deep within the brainstem there is a region called the periaqueductal gray, a dense cluster of neurons that plays a central role in pain suppression. When activated, the periaqueductal gray sends descending signals down through the spinal cord that suppress incoming pain signals at the gate. It is one of the most powerful pain-modulating systems in the body, and it achieves its effects primarily through a class of molecules you have almost certainly heard of: endorphins. Endorphins are the brain's endogenous opioids, molecules produced by the body itself that bind to the same receptors as morphine and other opioid drugs. They were discovered in the 1970s, and their discovery answered a question that had puzzled scientists for some time. Why does the brain have receptors perfectly shaped to receive opioid molecules? The answer was that the brain makes its own version of those molecules. It has its own built-in pain relief system, activated under the right conditions. Exercise releases endorphins. This is well established, and it is part of why sustained physical activity can produce a sense of well-being and ease that persists well after the exertion ends. But endorphins are also released in response to laughter, to genuine social connection, to music that moves you, to experiences of awe and beauty. The body is not stingy with its own pain relief chemistry. It makes it available through many doors. Alongside endorphins, the brain uses other chemical systems for pain modulation: serotonin, norepinephrine, and other neurotransmitters all play roles in the descending pathways that regulate pain. Many antidepressant medications, which work on these same chemical systems, have measurable pain-reducing effects independent of their effects on mood. This is not a coincidence. The systems are shared. But perhaps the most intriguing aspect of the brain's capacity for pain modulation is this: it can be activated by belief. What a person believes about their pain, what they expect, what meaning they assign to it, what they have been told about it, has real, measurable effects on the neurochemistry of their pain experience. This is the mechanism behind one of the most discussed and least understood phenomena in all of medicine: the placebo effect. The word placebo often carries a dismissive connotation in everyday language, as if something being a placebo means it is not real, not effective, somehow a trick. But this fundamentally misunderstands what is actually happening. A placebo is not the absence of an effect. It is a real biological effect generated by the brain in response to expectation, context, and belief. When a person receives a placebo treatment, a sugar pill, a saline injection, a sham procedure, and experiences genuine pain relief, their brain has produced that relief through real neurochemical means. Studies using brain imaging have shown that placebo analgesia involves actual activation of the same endorphin-releasing systems that opioid medications target. The periaqueductal gray activates. Endorphins are released. The descending pain suppression pathways engage. Real chemistry. Real neural activity. Real reduction in pain. The expectation of relief triggers the biological mechanisms of relief. The belief is not separate from the physiology. In this context, the belief is the physiology. And this effect is not small. In clinical trials, placebo treatments consistently produce significant pain reduction, often accounting for a substantial portion of the total effect seen in the active treatment arm. Some researchers estimate that across many types of pain, the placebo component of a treatment's effect may be larger than its direct pharmacological component. The belief the treatment generates may do as much work as the treatment itself. Positive beliefs more broadly, the belief that recovery is possible, that the pain is understandable and not permanent, that the body is fundamentally capable of healing, have measurable effects on pain intensity and on recovery outcomes. Patients who understand their pain in accurate, non-catastrophizing terms consistently do better than those who believe their pain signals ongoing damage or irreversible harm. This is not positive thinking as a platitude. It is positive thinking as a neurochemical intervention. What the mind believes, the brain implements. And now, with a clearer picture of how pain is generated, modulated, and shaped by the mind, it is worth stepping back and looking at the broader landscape of what pain actually is, in its many forms. Because pain is not one phenomenon. The word covers a family of experiences that share some features but differ significantly in their origins, their biology, their timelines, and what they require in response. Nociceptive pain is the most familiar. It is the pain that arises from direct tissue damage or the threat of it, the pain of a cut, a burn, a fracture, a bruise. It is the system working as designed, generating appropriate signals in response to appropriate stimulation. Generally, when the tissue heals, this type of pain resolves. It has a cause, a course, and an end. Inflammatory pain is related but distinct. It arises when the immune system's inflammatory response sensitizes the surrounding tissue, making an area hypersensitive as part of the healing and protection process. The deep ache of an infected joint, the tenderness around a healing wound, the diffuse soreness of a body fighting illness, these are all inflammatory pain in action. It is purposeful, but it can also overstay its welcome when inflammation becomes chronic. Neuropathic pain is different in kind. It arises from damage or dysfunction of the nervous system itself, not from the tissues the nerves serve, but from the nerves and the pathways themselves. A compressed nerve, a damaged spinal cord, the aftermath of certain viral infections, the complications of diabetes, these can all produce neuropathic pain. It tends to have a distinctive character, burning, shooting, electric, often described as lightning-like or searingly hot. It can be extraordinarily difficult to treat, because it does not respond well to the approaches that work for nociceptive pain. The source is not in the tissues. It is in the wiring. And there is a category that continues to be refined and debated in the field, but that represents a real and significant experience for millions of people: nosoplastic pain. This is pain that arises not from clear tissue damage, not from identifiable nerve damage, but from alterations in how the nervous system processes signals, the central sensitization we discussed earlier, the threshold lowering, the amplification of normally innocuous inputs. Fibromyalgia is perhaps the most recognized condition in this category. The pain is real. The suffering is real. But the source is in the altered processing of the system rather than in an identifiable structural problem. Each of these types of pain calls for different understanding and different approaches. Treating neuropathic pain the same way as nociceptive pain, or dismissing nosoplastic pain because no scan reveals a clear source, are errors that have caused tremendous unnecessary suffering. And then there is one of the stranger, more quietly startling behaviors that pain can exhibit: it can travel. Pain does not always stay where it originates. It can appear in locations that have no direct connection to the source of the problem, a phenomenon called referred pain. And it occurs because of the way sensory signals from different parts of the body converge on the same pathways in the spinal cord. When signals from multiple regions share the same spinal pathway, the brain sometimes cannot accurately determine which signal is coming from where. It makes its best guess, based on experience and prior learning, and it maps the pain to the location it considers most likely. But sometimes it guesses wrong. The most well-known example is the left arm pain of a heart attack. The heart and the left arm share overlapping nerve pathways in the spinal cord. When the heart is in distress and sending urgent signals, the brain sometimes maps that signal to the arm, a location it is more accustomed to associating with that particular type of signal. The heart is the source. The arm is where the pain appears. Referred pain from the diaphragm can appear in the shoulder. Referred pain from the lower back can appear in the hip or leg. Gallbladder pain can refer to the right shoulder blade. These are not random. They follow consistent patterns determined by shared neural anatomy, and once you understand that, the patterns begin to make a certain kind of sense. Pain, in all of its forms, in all of its behaviors, is a system doing its best to communicate something true and urgent. Even when it refers to the wrong location. Even when it overshoots and becomes chronic. Even when it amplifies beyond what is useful. It is always, at its root, an attempt to help. And understanding it, really understanding it, the way we have been moving through it tonight, is one of the quiet ways we can begin to work with it rather than simply against it. There is something deeply disorienting about referred pain when you first truly understand it. You have spent your entire life trusting that pain tells you where the problem is. You hurt your knee, and your knee hurts. You burn your hand, and your hand hurts. The location of the pain seems like the most basic, reliable piece of information the experience provides. And then you learn that this assumption, this foundational trust, is not always warranted. Pain can point to the wrong address. We touched on this briefly before, but it deserves to be understood more fully, because referred pain is not a rare anomaly. It is a regular feature of how the pain system works, and it has misled people and their doctors countless times throughout history. The reason referred pain exists comes back to the architecture of the spinal cord. Sensory nerves from different parts of the body do not each have their own private lane into the central nervous system. They converge. Multiple nerves from different regions feed into the same spinal segments, sharing pathways, overlapping their territories. The brain receives a signal through a shared channel and must decide where it originated. Most of the time, it makes a reasonable guess. But when an unusual source, like the heart, or the gallbladder, or the diaphragm, sends a signal through a pathway the brain more commonly associates with somewhere else, the brain can map the pain to the more familiar location. This is why a problem with the gallbladder can produce pain felt under the right shoulder blade. Why irritation of the diaphragm can feel like shoulder tip pain. Why a kidney issue can appear as a deep ache in the lower back or flank. Why certain types of neck dysfunction can produce pain felt in the arm or hand. The original problem and the felt pain can be separated by significant physical distance, connected only by the shared pathways in the nervous system and the brain's best, imperfect attempt at interpretation. For people living with unexplained pain in a location where nothing appears to be wrong, understanding referred pain can be genuinely illuminating. The absence of a structural problem in the painful area is not proof that the pain is imagined. It may simply mean that the source lies elsewhere, quietly sending signals through a shared channel, waiting to be correctly identified. But if referred pain is disorienting, what comes next in our story is something altogether more remarkable. Phantom pain. When a limb is amputated, when an arm or leg is surgically removed, or lost to injury, many people continue to feel that limb. Not just a vague sense of its absence. They feel its precise shape, its temperature, its position in space. They can feel the fingers of a hand that no longer exists, bent into a fist. They can feel the toes of a foot that is gone, pressing against a floor that cannot be reached. And in many cases, around 60 to 80% of amputees experience this. They feel pain in that absent limb. Pain that is undeniably real, neurologically generated, sometimes severe, and located in something that is no longer there. Phantom pain was poorly understood for most of medical history, and for a long time it was not taken seriously, treated as a psychological curiosity, a grief response, something that would resolve with time and acceptance. But it is not psychological in the dismissive sense. It has a clear neurological basis, and understanding it reveals something fundamental about where pain truly lives. The key lies in the brain's body map. Recall the somatosensory cortex, the region of the brain that contains a detailed representation of the entire body surface, each area mapped to a corresponding strip of cortex. When a limb is amputated, the sensory input from that limb suddenly stops arriving. The corresponding region of the cortex goes quiet, deprived of the signals it has been receiving for a lifetime. But the cortical representation itself does not immediately disappear. It remains. And in its deprivation, it becomes unstable. Neighboring regions of the cortex, now carrying more traffic, can begin to encroach into the silenced territory. The cortex reorganizes, a process called cortical remapping, and this reorganization can produce spontaneous activity in the region that once represented the missing limb. The brain, drawing on its map, interprets this activity as coming from where it always came from. And so the missing limb seems present. And when that activity carries the signature of a pain signal, the missing limb seems to hurt. The brain generates the pain from its own map, in the absence of any peripheral input. This is perhaps the most vivid possible demonstration of something we have been building toward throughout this entire journey. Pain is made in the brain. Not detected, not transmitted, not passively received, made. The brain constructs the experience using its own internal representations, and those representations can operate independently of the physical body they were built to represent. Phantom pain has also produced one of the more elegant and ingenious treatments in modern pain medicine, the mirror box, developed by neuroscientist Vilayana Ramachandran in the 1990s. The device is simple, a box with a mirror positioned so that when the patient places their remaining limb inside and looks at the reflection, the brain perceives it as the missing limb. The patient can then watch in the mirror as they move the existing hand or foot, and the brain, seeing what appears to be the absent limb moving freely, can sometimes release a frozen phantom, relax a cramped one, and provide significant relief from pain. A mirror. A reflection. And the brain, offered visual evidence that contradicts its frozen map, can update its representation and dissolve pain that had no physical source. There is something almost poetic in that. The brain created the pain with its map. And a piece of reflective glass could help revise the map and undo it. This understanding of phantom pain has also opened new possibilities in prosthetics. Modern prosthetic limbs that provide sensory feedback, that can send signals back to the residual limb or directly to the nervous system, telling the brain that something is being touched, that pressure is being applied, that the prosthetic is moving, have shown promise not just in improving function but in reducing phantom pain. When the brain receives input through a prosthetic that partially restores the sensory conversation it lost, the cortical map can stabilize. The phantom quiets. Pain that arose from sensory deprivation can ease when the deprivation is addressed. The brain is not a passive sufferer. Even in its most difficult states, it retains the capacity to adapt. And here is one final fact about the brain and pain that tends to stop people in their tracks when they first hear it. The brain itself, the organ that generates pain, that processes every signal, that constructs every ache and throb and burning sensation, does not have nociceptors. The brain tissue has no pain receptors of its own. It cannot feel pain directly. Surgeons can operate on the surface of the brain in conscious patients, as has been done in certain types of brain surgery where patient feedback is valuable, and the patient feels no discomfort from the manipulation of brain tissue itself. The experience of being touched there, cut there, is absent. What does hurt in and around the head, in headaches, in migraines, in surgical incisions, are the structures surrounding and supplying the brain, the meninges, the membranes that wrap the brain and spinal cord, the blood vessels in and around the brain, the scalp, the muscles of the head and neck. These structures have nociceptors in abundance. But the organ itself, the 3 pounds of tissue doing all of this extraordinary work, feels none of it. There is something quietly vertiginous about that. The creator of all pain experience is immune to the very thing it creates. Now, let us turn to what happens when the system that was built to protect you begins, in a sense, to work against you. Acute pain has a logic that is easy to follow. Something happens, the system responds, and when the event resolves, the pain resolves with it. The system did its job, the threat passed, and the alarm was switched off. Clean, purposeful, finite. But for hundreds of millions of people around the world, pain does not switch off. It persists beyond any clear injury. It outlasts the healing of tissues. It continues in the absence of ongoing damage, or in the presence of conditions that would not seem to justify its severity. It becomes chronic. For most of medical history, chronic pain was understood as a symptom, the ongoing expression of an underlying problem that simply had not resolved yet. The assumption was that if you could find and fix the underlying cause, the pain would go away. And sometimes that is true. But for a significant proportion of people living with chronic pain, the underlying cause is resolved, or cannot be found, or was never clearly identified, and the pain continues anyway. Modern pain science has arrived at a conclusion that is both clarifying and deeply important. Chronic pain is often not a symptom of a disease. It is the disease. A distinct condition in its own right, with its own biology, its own mechanisms, its own brain changes, requiring its own forms of treatment rather than simply more investigation in search of an elusive structural cause. What has happened in chronic pain, at the neural level, is that the protective system has malfunctioned. The sensitization that was supposed to be temporary has become permanent. The alarm that was supposed to turn off has become stuck in the on position. And a system that was built for short-term protection is now operating continuously, generating ongoing suffering with no resolution in sight. The pain system stays sensitized. Central sensitization, which we explored earlier in the context of repeated signals lowering the threshold, can become self-sustaining. The pathway is so deeply carved, the system so thoroughly recalibrated towards sensitivity, that it no longer requires significant external input to remain active. The nervous system has learned to be in pain. And like many learned things in the nervous system, this learning is resistant to simply being told to stop. The brain can generate pain entirely on its own, without any incoming signals from the periphery, once the system has reorganized sufficiently around that chronic state. This is why blocking the peripheral nerves, the original source of the signal, sometimes fails to relieve chronic pain. The pain is no longer primarily peripheral. It has moved inward. It has become, in a meaningful sense, a brain phenomenon, sustained by the altered state of the central nervous system rather than by ongoing damage in the tissue. This has profound implications for how chronic pain should be understood and treated. Approaches focused entirely on the body part that hurts, repeated imaging, repeated local injections, surgical interventions aimed at structural abnormalities, often fail in chronic pain precisely because the primary problem is no longer in that body part. It is in the system that processes signals from that body part. Treating the periphery when the central nervous system is reorganized is like repeatedly replacing a smoke detector's battery when the alarm software itself has become corrupted. And then there is a dimension of chronic pain biology that has only become fully appreciated in recent years, one that has shifted the scientific conversation significantly and that involves cells most people have never heard of. Glial cells. For most of neuroscience history, glial cells were considered the support staff of the nervous system. Their name comes from the Greek word for glue. They were thought of as the structural scaffolding that held neurons in place, fed them, cleaned up after them. Passive. Secondary. The background against which the real neural action occurred. This view was wrong. Or at least, dramatically incomplete. Glial cells, particularly a type called microglia, and another called astrocytes, are now understood to play an active, significant role in the processing and amplification of pain. And in chronic pain states, their role may be central. Microglia are the immune cells of the central nervous system. They are constantly patrolling, scanning the neural environment for signs of damage, infection, or abnormality. When they detect a threat, they activate. And when they activate in the context of persistent pain signaling, they release a cascade of inflammatory molecules, cytokines, prostaglandins, and other chemical signals, directly into the environment of the neurons around them. These molecules sensitize the neurons they surround. They lower the neurons' thresholds. They make the pain pathways more reactive, more easily activated, more prone to firing in response to minimal input. And they can keep doing this long after the original trigger has resolved, maintaining a state of neural inflammation that perpetuates the pain experience even in the absence of ongoing tissue damage. Astrocytes, the other major glial type involved, play a different but complementary role. They regulate the chemical environment around synapses, controlling the concentration of neurotransmitters, modulating the signaling between neurons. In chronic pain states, astrocytes can alter their function in ways that effectively turn up the gain on pain circuits, making them more efficient at transmitting and amplifying pain signals. What this means is that chronic pain has an inflammatory component that is not in the tissues of the body, but inside the nervous system itself. Neuroinflammation, inflammation of the central nervous system, is now recognized as a key driver of chronic pain conditions. And it operates through cells that were, until relatively recently, not even considered relevant to the pain story. This discovery has opened new avenues for treatment. Researchers are exploring whether targeting glial activation, quieting the neuroinflammatory response in the central nervous system, might offer relief for people with chronic pain conditions where conventional treatments have failed. Early results are promising enough that this has become one of the most active areas of pain research in the world. The picture that emerges from all of this, from referred pain and phantom pain, from central sensitization and glial activation, from brains that generate pain without peripheral input and systems that learn to stay in alarm, is one of extraordinary complexity. A system that was designed with breathtaking precision for short-term protection, revealing its fragility when asked to operate over the long term. Chronic pain is not weakness. It is not catastrophizing. It is not a failure of will or attitude. It is a nervous system that has adapted to its conditions, changed itself, as nervous systems do, and gotten stuck in a configuration that causes suffering rather than preventing it. And the people living inside that experience deserve, above everything else, to be believed, to be understood. and to be met with the full weight of scientific seriousness that a condition of this complexity demands. One of the more unsettling discoveries in modern pain science is how thoroughly chronic pain can rewrite the nervous system over time. Not metaphorically, not as a way of describing how it feels, but literally, at the level of individual neurons, individual synapses, individual genes. The biology of the nervous system changes in response to persistent pain in ways that are measurable, documentable, and in some cases visible on brain scans. The system that was built to protect you can, under the sustained pressure of chronic pain, transform itself into something that perpetuates suffering. And understanding how that transformation happens is one of the most important frontiers in pain medicine today. Let us begin with something that feels almost paradoxical. Under normal circumstances, the fibers that carry touch signals and the fibers that carry pain signals are different. They serve different purposes, they connect to different pathways, and they produce different experiences. Touch is touch. Pain is pain. The two are processed through largely separate channels. But in chronic pain states, this separation can break down. When an area of the body is persistently sensitized, when inflammation or nerve damage has kept the nociceptive system in a heightened state for an extended period, something remarkable can happen at the level of the spinal cord. The touch fibers, called A-beta fibers, which normally carry only innocuous sensory signals, begin to make new connections. They sprout, reaching into the pain-processing layers of the spinal cord, layers they do not normally innervate. And in doing so, they begin to feed their signals into the pain pathway. The result is a phenomenon called allodynia, pain produced by stimuli that are not, under any normal circumstances, painful. A light stroke across the skin. The gentle pressure of clothing. A warm hand resting on an arm. These touch signals, traveling through fibers that were never built to cause pain, are now routing through a pain-sensitized system and emerging as genuine, sometimes severe pain. People with conditions like fibromyalgia, complex regional pain syndrome, or certain neuropathic pain disorders often experience allodynia. When they describe being unable to tolerate the feeling of fabric against their skin, or finding a gentle touch unbearable, they are not exaggerating and they are not being fragile. Their nervous system has structurally changed in a way that has turned ordinary touch into a pain trigger. The wiring has been rewired. At the cellular level, a key player in this sensitization process is a receptor called the NMDA receptor, short for N-methyl-D-aspartate. NMDA receptors are found on neurons throughout the central nervous system, and under normal circumstances they play a role in learning and memory, helping neurons strengthen connections through experience. But in the context of persistent pain signaling, these same receptors drive a form of learning that nobody would choose. When pain signals arrive at the spinal cord repeatedly and with sufficient intensity, the NMDA receptors on the receiving neurons become increasingly activated. Their activation leads to a cascade of changes inside the neuron, changes that lower the neuron's threshold for firing, increase its responsiveness, and enhance its ability to transmit pain signals upstream. This is called long-term potentiation of pain pathways, the same cellular mechanism that underlies learning and memory, applied to the circuits of pain. the neuron has, in essence, learned to be better at pain. It fires more easily, more intensely, in response to less. And here the story deepens further. Because the changes do not stop at the receptor level. They reach all the way down into the nucleus of the cell, into the DNA itself. Persistent pain changes gene expression in the neurons of the pain pathway. This does not mean the DNA sequence changes, the underlying genetic code remains the same. But which genes are turned on and which are turned off, the pattern of gene expression, shifts in response to sustained pain signals. Neurons begin producing different proteins, more of certain ion channels that make the cell membrane more excitable, different configurations of receptors that alter the cell's sensitivity, changes in the enzymes that regulate neurotransmitter production. The cell has been reprogrammed by experience. It is not the same cell it was before the chronic pain began. Its molecular identity has been altered by the pattern of signals it has been asked to process. This is one of the reasons why chronic pain can be so resistant to treatment. The changes that have occurred are not superficial adjustments to a system that is otherwise intact. They are deep, molecular alterations to the fundamental behavior of the cells in the pain pathway. Addressing them requires not just blocking a signal but, in some sense, convincing a transformed system to transform again, in a different direction. There's also a more destructive side to what chronic pain does to neurons over time. In some chronic pain conditions, sustained neural inflammation and excessive neural activity contribute to actual degeneration, the loss of neurons in specific regions. Studies of people with long-term chronic pain have found measurable reductions in gray matter density in areas of the prefrontal cortex, the thalamus, and other regions involved in pain processing and emotional regulation. This is not simply the brain changing its processing patterns. This is the chronic activation of pain circuits, combined with neuroinflammation and the toxic effects of excessive glutamate activity, contributing to cell damage over time. Pain, at its most persistent and severe, can literally cost the brain neurons, reducing the mass and connectivity of regions that are essential not just for pain processing, but for attention, decision-making, emotional regulation, and the quality of daily life. This finding carries considerable weight. It suggests that effective treatment of chronic pain is not merely a matter of comfort or quality of life, though those would already be sufficient justification. It is also a matter of preserving the structural integrity of the brain itself. And yet, and this must be said with equal clarity, the brain that can lose volume under chronic pain is also capable of recovering it. Studies have shown that effective pain treatment, when it succeeds in significantly reducing chronic pain, is associated with partial recovery of gray matter in the affected regions. The brain regains some of what was lost. Neuroplasticity, which contributes to the problem, is also part of the solution. Now let us look at what chronic pain does to the broader architecture of the brain over time, beyond the molecular and cellular level, at the level of whole systems and their relationships. One of the most significant changes involves the dopamine system. Dopamine is perhaps the most widely discussed neurotransmitter in popular culture, usually in the context of reward and pleasure. But its role is more precisely described as motivational salience. It is the chemical signal that marks things as important, that drives the pursuit of outcomes, that encodes the value of experiences and behaviors. Under normal circumstances, the dopamine system works in close relationship with the pain system. Acute pain triggers dopamine release in certain pathways, part of the motivational response to threat, driving the organism to act, to escape, to resolve the situation. And once the pain resolves, the dopamine system participates in the sense of relief, in the rewarding experience of a threat having passed. In chronic pain, this relationship becomes distorted. The dopamine system is altered, its baseline activity changes, its responsiveness to reward decreases, and its relationship to the pain pathway shifts in ways that are still being fully mapped. People living with chronic pain often report a loss of pleasure in things that once brought enjoyment, a flattening of the capacity for positive experience, a difficulty accessing the sense of reward that normally motivates and sustains engagement with life. This is not simply depression as a secondary consequence of living with pain, though depression is genuinely common in chronic pain populations and the relationship is bidirectional. It is a direct neurochemical consequence of what chronic pain does to the dopamine system over time. The reward circuitry has been rewired around the constant presence of pain, and finding pleasure in ordinary things becomes genuinely, neurologically harder. The brain's reward system more broadly, the interconnected networks that process value, motivation, anticipation, and satisfaction, is affected in chronic pain in ways that go beyond dopamine alone. The prefrontal cortex, which provides top-down regulation of both pain and emotion, becomes less effective at modulating the signals coming from the amygdala and the pain processing regions below. The balance between bottom-up pain signals and top-down regulatory control shifts. Pain becomes harder to regulate, harder to contextualize, harder to place in perspective. Not because the person is failing at something, but because the circuits that would enable that regulation have been altered by the chronic experience they have been asked to handle. There is another change that occurs in chronic pain, one that, when researchers first documented it, seemed almost too strange to be true. The brain's internal map of the body becomes distorted. Recall the somatosensory homunculus, the brain's detailed representation of the body surface, laid out across the cortex. This map is not permanently fixed. It is plastic, it can be updated, refined, and reshaped by experience. When a part of the body is used intensively, its representation on the map expands. When input from a region is reduced, its representation can shrink or be encroached upon by neighbors. In chronic pain conditions, particularly those involving a specific body region, the cortical representation of that region can become blurred and distorted. The precise mapping that normally allows the brain to accurately locate sensation begins to lose its resolution. The borders between adjacent regions blur. The brain's GPS for the painful area becomes, in a sense, smeared. One of the clinical consequences of this distortion is something that has been documented in conditions like chronic low back pain and complex regional pain syndrome, left-right confusion. People with significant cortical reorganization around a chronically painful region sometimes lose accuracy in distinguishing left from right, not conceptually, but in their sense of their own body in space. Tasks that involve mentally rotating or imagining movements of the painful limb become difficult in ways that have nothing to do with the limb's physical capability. The brain's map of that body part has become sufficiently disrupted that spatial operations involving it are impaired. This finding has led to treatments based on cortical remapping, approaches like graded motor imagery, which gradually retrains the brain's representation of the affected area through visualization and imagination before any physical movement is attempted. The goal is to restore the map before restoring the movement, to give the brain accurate spatial information about the affected regions so that the disorganized representation can reorganize towards something more precise. It is a striking approach because it does not touch the body part at all in its early stages. It works entirely through the brain. And it has shown genuine effectiveness in some of the most difficult chronic pain conditions. Over time, the pain responses themselves adapt, not toward resolution, but toward a kind of grim equilibrium. The system recalibrates around pain as its new baseline. The baseline level of neural activity in the pain processing networks rises. The fluctuations that occur above that baseline, the spikes of heightened pain in response to triggers, are processed against a background that is already elevated. Relief, when it occurs, is measured not from zero but from a higher starting point. The entire scale has shifted. This adapted state is one reason why people who have lived with chronic pain for years can become difficult to treat even with interventions that might work well earlier in the condition's course. The window has not just moved, the architecture of the room has changed. The brain that is receiving treatment is not the same brain that first developed the pain. It is a brain that has been reorganized, resensitized, structurally altered, and functionally recalibrated around a sustained pain experience. None of this is the person's fault. None of it reflects a character failure or a lack of effort. It is biology responding to biology, a nervous system doing what nervous systems do, which is learn from and adapt to their experience. The tragedy of chronic pain is not that the system failed. It is that the system worked exactly as designed, in circumstances that turned its own intelligence against it. And yet, even in the most reorganized, most sensitized, most chronically altered nervous system, the capacity for change remains. Neuroplasticity is not only the mechanism of the problem. It is the foundation of every hope for improvement. The brain that changed in one direction under pressure retains, in principle, the ability to change again. That possibility, quiet, biological, grounded in the same science that describes the damage, does not make chronic pain any less real, any less difficult, or any less deserving of serious medical attention. But it is real. And it matters. The brain is never entirely finished becoming what it might yet be. There is a version of pain science that lives entirely inside the nervous system, in neurons and receptors and cortical maps and glial cells. And that version is real and important and worth understanding deeply. But pain does not live only inside the nervous system. It lives inside a whole body, in a whole life, embedded in a physical environment that changes by the hour and a biological landscape that is shaped by sleep, by food, by hormones, by the state of organs that seem, at first glance, to have nothing to do with pain at all. The factors that influence how much pain a person feels on any given day extend far beyond what is happening at the injury site or inside the spinal cord. They include things as seemingly ordinary as the weather outside, the position they slept in, what they have been eating, and the state of their digestive system. These are not soft variables at the edges of the real science. They are part of the biology, woven into the same systems we have been tracing throughout this journey. Let us begin somewhere almost everyone has encountered, even if they have never had a name for it. Weather affects pain. This is one of those observations that sits in an uncomfortable space between folk wisdom and formal science. Grandparents who predicted rain by the ache in their joints were often dismissed as fanciful. But the evidence, accumulated carefully over decades, suggests they were onto something real. The leading explanation involves barometric pressure, the pressure of the atmosphere pushing down on the body. On a clear, high-pressure day, that atmospheric pressure helps to compress the tissues around joints, providing a kind of external support to the structures within. When a low-pressure system moves in, bringing clouds, rain, or storms, that external compression decreases. The tissues around joints have slightly more room to expand. For joints that are already inflamed or damaged, this small change in mechanical conditions can be enough to increase the sensitivity of the local nociceptors and produce a noticeable increase in pain. Cold temperature adds another layer. Cold causes the muscles and connective tissues around joints to stiffen, reducing their flexibility and increasing the mechanical stress on the joint surfaces during movement. The blood vessels and peripheral tissues constrict in cold, reducing circulation to the extremities. And cold temperatures may directly increase the sensitivity of certain nociceptors, particularly those tuned to temperature and mechanical stimulation. The result is that people with joint conditions, old injuries, or neuropathic pain often find winter significantly harder than summer. Humidity plays a role as well, though the mechanisms are less clearly understood. High humidity is associated with increased pain reports in several conditions, possibly because of its effects on tissue hydration, nerve conduction, or inflammation. What is clear is that the relationship between pain and weather is not imagined. It is a real biological phenomenon, mediated through measurable physical mechanisms, that varies between individuals based on their specific conditions and the particular sensitivities of their nervous systems. The environment inside the sleeping body matters just as much as the environment outside it. Sleep posture, the position in which the body rests through the night, can have significant and direct effects on pain, and the relationship runs in both directions. Poor sleep posture can create or worsen pain. An existing pain can disrupt sleep, preventing the body from reaching the restorative stages it needs. The two feed each other in a cycle that, once established, can be remarkably difficult to break. During sleep, the spine needs to maintain something close to its natural alignment, the gentle curves that distribute mechanical stress evenly across the discs and joints. When posture collapses those curves, when the head is pushed too far forward, when the lumbar spine loses its support and sags, when a shoulder is compressed under the body's weight for hours, the sustained mechanical stress accumulates. Muscles that are held in shortened or lengthened positions throughout the night wake stiff and reactive. Joints that have been compressed or distorted for hours begin the day in a sensitized state. The mattress and pillow matter in this context not as luxury items but as biomechanical equipment, tools that either support the body's natural architecture through the hours of stillness or allow it to deform. People who wake with pain that eases gradually over the course of the morning are often experiencing the delayed resolution of postural stress accumulated during the night. The tissue was not damaged, but it was held in an uncomfortable configuration for long enough that the local nociceptive system registered it as a problem. Beyond posture, sleep itself, the quality and duration of it, is one of the most powerful modulators of pain sensitivity that exists. Even a single night of poor sleep has been shown in controlled studies to measurably lower pain thresholds the following day. Sleep deprivation increases inflammation, dysregulates the descending pain inhibition pathways, reduces the efficacy of the brain's own endorphin systems, and elevates stress hormones, all in the direction of greater pain sensitivity. Chronic sleep disruption, as experienced by many people with chronic pain conditions, creates a state of sustained neurological vulnerability. The system that should be dampening pain signals is running on diminished resources, and the threshold for pain experience drops accordingly. Sleep and pain are not separate problems to be addressed independently. They are the same problem, wearing two different faces. Then there is nutrition, and specifically, the effect of nutrient deficiencies on the pain system. Vitamin D is perhaps the most well-documented nutritional factor in pain. It is not conventionally thought of as a pain-related nutrient, but its receptors are found throughout the nervous system, including in regions involved in pain modulation. Deficiency in vitamin D has been associated in multiple studies with increased musculoskeletal pain, lower pain thresholds, and poorer outcomes in people with chronic pain conditions. In populations where vitamin D deficiency is widespread, which, in many parts of the world with limited sun exposure, is a significant proportion of the population, this represents a modifiable contributor to pain that is often overlooked entirely. Magnesium is another mineral whose deficiency has direct relevance to pain. Magnesium plays a role in regulating NMDA receptor activity, the very receptors we discussed earlier as central drivers of central sensitization. Under normal circumstances, magnesium partially blocks NMDA receptors, limiting their ability to amplify pain signals. When magnesium levels are low, this natural inhibition is reduced. The NMDA receptors become more active. The sensitivity of the pain pathways increases. For people with muscle cramps, tension headaches, or conditions involving central sensitization, magnesium deficiency may be a quiet amplifier of the problem. Omega-3 fatty acids, found in oily fish, flaxseed, and certain other foods, have anti-inflammatory properties that operate at a cellular level. They influence the production of prostaglandins and other inflammatory mediators, shifting the balance away from the pro-inflammatory molecules that sensitize nociceptors and toward molecules that resolve inflammation. Diets chronically low in omega-3s and high in omega-6 fatty acids, a pattern that characterizes much of modern industrial eating, tip this balance in the direction of greater inflammation and potentially greater pain sensitivity. The body runs on chemistry. and the chemistry it has available to work with is largely determined by what it has been given to work with. Now we arrive at one of the more surprising entries in the story of pain, a system that most people would not think to connect to their nervous system at all. The gut. The relationship between the gut and the experience of pain is one of the most rapidly developing areas in pain research, and what has emerged over the past decade or so has fundamentally changed how scientists think about chronic pain conditions that were previously poorly understood. The gut contains its own extensive nervous system, the enteric nervous system, sometimes called the second brain, with more neurons than the spinal cord. It is in constant bidirectional communication with the brain through the vagus nerve, through hormonal signals, and through the immune system. And it hosts a vast ecosystem of microorganisms, the gut microbiome, whose influence extends far beyond digestion. The gut microbiome produces neurotransmitters. It influences the production of serotonin. A significant portion of the body's total serotonin is produced in the gut, not the brain. It regulates immune function. It influences the levels of inflammatory molecules circulating in the bloodstream. And through all of these channels, it exerts a meaningful influence on the sensitivity and behavior of the central nervous system. When the gut microbiome is disrupted, through poor diet, antibiotic use, chronic stress, or illness, the balance of its microbial population shifts. Species that produce anti-inflammatory compounds may decline. Species associated with increased intestinal permeability, the condition sometimes described as leaky gut, may proliferate. The intestinal lining becomes more permeable, allowing bacterial fragments and inflammatory molecules to enter the bloodstream and reach the systemic circulation. These circulating inflammatory signals do not stay in the body periphery. They cross into the central nervous system through a variety of routes, where they can activate the glial cells we discussed earlier, the microglia and astrocytes, triggering the neuroinflammatory response that amplifies pain signaling. Gut inflammation, in this way, can drive central sensitization through the back door. The problem starts in the intestine. The amplification happens in the brain. This connection between gut health and chronic pain has been documented in a growing number of conditions: fibromyalgia, irritable bowel syndrome, migraine, rheumatoid arthritis, and others. People with these conditions show consistent differences in their gut microbiome composition compared to people without them. Whether the gut changes come first and contribute to the pain, or whether the pain and its associated stress alter the gut, or whether both are true, the relationship is bidirectional, mutually reinforcing, and clinically significant. Dietary approaches aimed at reducing intestinal inflammation, reducing ultra-processed foods, refined sugars, and pro-inflammatory fats while increasing fiber, fermented foods, and diverse plant-based inputs, are increasingly being studied as adjuncts to pain treatment, not as alternative medicine but as legitimate biological interventions targeting a real pathway. The modern lifestyle has also created new patterns of pain that would have been largely unfamiliar to human bodies living even a century ago. The sustained static postures demanded by screen-based work, the loss of diverse physical movement across daily life, the shift toward highly processed diets, the epidemic of sleep disruption from artificial light and screen exposure, the chronic low-grade stress of information overload, All of these are biological inputs into systems that were not designed for them. Pain that arises from hours of motionless sitting, from the unrelieved forward flexion of desk work, from the absence of the varied physical loading that kept connective tissue healthy throughout most of human evolution. This is not a character failing or a consequence of softness. It is a biological mismatch between an ancient body and a very new set of demands. The systems evolved for movement, for varied loading, for natural light cycles and genuine rest, are being asked to operate under conditions that systematically undermine their function. Understanding this does not make modern life's demands disappear. But it does clarify what the body needs, and why meeting those needs is not optional if the goal is to live with less pain. Now let us turn to some of the more remarkable variations in how pain is experienced across different people and different biological states. There exist, in the human population, individuals who feel no pain at all. This condition, congenital insensitivity to pain, is rare, and it might sound, at first consideration, like an enviable gift. But people who live with it uniformly describe it as a serious and life-threatening medical condition. Without pain, the body's warning system is absent. Children with congenital insensitivity to pain bite their tongues and fingers without knowing it, break bones without realizing it, develop severe joint damage from repeatedly overloading injured structures that send no distress signal. They cannot tell when they have appendicitis, or when a wound is becoming infected, or when they are developing a pressure ulcer from sitting in one position too long. They must learn to perform systematic body checks, visual inspections of their entire skin and joint surfaces, because the internal alarm system that would alert the rest of us simply does not exist for them. The condition arises from mutations in specific genes, often the gene encoding a sodium channel called NaV1.7, which plays a critical role in the transmission of signals from nociceptors. Without functional NaV1.7, nociceptor signals cannot be effectively transmitted, and pain experience does not occur. This gene, and the protein it encodes, has become one of the most intensively studied targets in pain medicine, not to eliminate pain entirely, but to understand the molecular machinery well enough to modulate it more precisely in people for whom it has become pathological. The existence of congenital insensitivity to pain is a vivid reminder of something worth sitting with. Pain, as difficult and as unwelcome as it so often is also one of the most important features of a functional human body. Its presence is a sign that the system is working. Its complete absence is a medical emergency in slow motion. Hormones add another dimension of variation to the pain experience, one that is particularly significant and that has historically been underappreciated in clinical practice. Estrogen, progesterone, and testosterone all have measurable effects on the pain system. Estrogen, in particular, appears to have complex and sometimes paradoxical influences on pain sensitivity. At certain levels, it can have pain-dampening effects. Estrogen interacts with the opioid system, with serotonin pathways, and with inflammatory cascades in ways that can reduce nociceptive sensitivity. But fluctuating estrogen levels, as occur during the menstrual cycle, the perimenopause, and the postmenopause, are associated with increased pain sensitivity and with the onset or worsening of various pain conditions. This is why many pain conditions show a pronounced sex difference in their prevalence: fibromyalgia, migraine, temporomandibular joint disorders, irritable bowel syndrome, and several autoimmune pain conditions all occur significantly more frequently in women. For a long time, this disparity was attributed to psychological factors. But the hormonal biology offers a more complete and more respectful explanation. The female hormonal environment, across the reproductive lifespan, creates conditions in the pain processing system that are genuinely different from those in the male hormonal environment. These differences are biological, not psychological, and they deserve to be taken seriously in both research and clinical practice. The fluctuation of estrogen and progesterone across the menstrual cycle produces measurable changes in pain threshold. In the follicular phase, rising estrogen is generally associated with lower pain sensitivity. In the late luteal phase, when both estrogen and progesterone are declining in the days before menstruation, pain sensitivity tends to be highest. Women with dysmenorrhea, painful menstruation, experience this as something far more than ordinary cramping. The prostaglandin release that drives uterine contractions also drives systemic inflammation and heightened nociceptive sensitivity throughout the body. Testosterone, in contrast, generally has pain-dampening properties. Higher testosterone levels are associated with higher pain thresholds. The decline of testosterone with age in men, and the very different testosterone environment in women, contributes to the pattern of sex differences in pain that is observed across populations and across the lifespan. Diabetes introduces yet another form of pain variation, and one that affects an enormous number of people worldwide. Diabetic peripheral neuropathy arises from the effect of chronically elevated blood glucose on the peripheral nerves. High glucose levels are toxic to nerve cells over time, damaging the myelin sheaths that insulate nerve fibers, disrupting the cellular machinery within neurons, and impairing the microcirculation that supplies the nerves with blood and oxygen. The result, in many people with long-standing diabetes, is a progressive deterioration of peripheral nerve function, beginning in the extremities, typically the feet and lower legs, and spreading inward over time. The pain of diabetic neuropathy has a distinctive character that reflects the nature of the underlying damage. Burning, electric, shooting. Often described as walking on broken glass or having the feet wrapped in fire. At the same time, the damaged nerves may also lose their ability to carry protective signals accurately, meaning that an area of the foot that screams with burning pain may simultaneously have reduced sensation for pressure and temperature. The nociceptive system is misfiring, generating pain spontaneously, while the protective sensory function it was originally built to provide is compromised. This combination, pain without protection, is one of the cruelest aspects of diabetic neuropathy, and it underlies the serious foot complications that remain one of the condition's most significant consequences. What all of these variations, across weather, sleep, nutrition, gut health, hormones, genetics, and systemic disease, share as a common truth: the pain system is not an isolated mechanism. It is an expression of the whole body. It reflects everything that is happening across every system, every organ, every chemistry. It is sensitive to the state of the gut, the level of hormones, the composition of the microbiome, the quality of sleep, the amount of vitamin D in the blood, the barometric pressure in the air outside. To understand pain fully is to understand the body as an integrated whole, not a collection of separate parts but a single, deeply interconnected system, in which everything touches everything else, and in which the experience of pain is always the product of the entire person, not just the part that hurts. Pain does not stay the same across a lifetime. The body that experiences pain at 20 is a genuinely different biological system from the body that experiences it at 50 or 70. The nervous system ages. The tissues age. The hormonal environment shifts. The accumulated history of injuries, illnesses, stresses, and recoveries leaves its marks on the pain processing architecture in ways that gradually change both the frequency and the character of what is felt. In early life, the pain system is still calibrating. Infants and young children have pain systems that are not yet fully organized. The descending inhibitory pathways that allow the brain to dampen incoming signals are still developing, which is one reason young children often seem to experience pain more intensely and with less ability to modulate it than adults. The cortical regions that provide top-down regulation of the emotional response to pain are still maturing. The experience is real and significant, and it deserves to be taken seriously, even when, perhaps especially when, the child cannot yet articulate it clearly. As the nervous system matures into adulthood, the pain system reaches its fullest functional capacity. The inhibitory pathways are well developed. The cortical regulation is at its most efficient. The balance between amplification and dampening is, for most people, at its most favorable. This does not mean pain is mild or insignificant in adulthood. The chronic pain epidemic that affects hundreds of millions of working-age adults is evidence enough to the contrary. But the tools available to the brain for managing and modulating pain are at their most complete. With advancing age, the picture changes again, and it changes in multiple directions simultaneously. On one hand, some aspects of peripheral nociception become less sensitive with age. The density of certain nociceptors in the skin decreases over time. Nerve conduction velocity slows. The first sharp signal, the fast A-delta fiber response, can become somewhat blunted. This might seem like a protective change, and in some narrow sense it is. But it comes alongside a deterioration in the descending inhibitory systems that dampen pain centrally. The brain's ability to suppress incoming pain signals becomes less efficient. The endogenous opioid system becomes less responsive. The periaqueductal gray, with its powerful pain-suppressing circuitry, operates with reduced effectiveness. The net result, for many older adults, is a pain experience that may lack some of the sharp immediacy of younger pain, but that becomes more persistent, more diffuse, and harder for the brain to regulate. Chronic pain is significantly more prevalent in older populations, and the co-occurrence of multiple pain-generating conditions, osteoarthritis, spinal degeneration, peripheral neuropathy, post-surgical changes, creates a complex, overlapping picture that is genuinely more challenging to manage than a single well-defined condition in a younger person. There is also the matter of cognitive change. The prefrontal cortex, which plays such an important role in the top-down regulation of pain, in contextualizing it, in applying meaning to it, and dampening the emotional reactivity the amygdala brings, is one of the brain regions most affected by normal cognitive aging. As prefrontal function declines, the regulatory control it exerts over the pain processing system can weaken. The alarm becomes harder to turn down. And then there is the accumulated weight of history. An older body carries decades of experience with pain, injuries healed and unhealed, sensitized pathways, altered maps, memories and expectations built up over a lifetime. The nervous system of someone in their 70s has been shaped by everything that came before. It is not simply an aged version of the young system. It is a system that has been continuously rewritten by its own experience. Understanding this helps explain why pain management in older adults requires particular sensitivity and individualization. A one-size approach cannot account for the biological reality of a system that has spent decades becoming uniquely itself. But here, having traced pain from its molecular origins to its most complex expressions across a lifetime, it is worth pausing to ask a question that sits beneath all of the science. Why? Why does pain exist at all? What is the purpose of a system so elaborate, so costly, so capable of producing such profound and sustained suffering? The answer, at its simplest, is survival. But the simplicity of that answer conceals a depth worth exploring. Pain drives action. This is its most fundamental purpose, not to inform in a passive sense, but to compel. Pain is not a notification. It is a command. It is the nervous system's most powerful tool for changing behavior, and it was shaped by hundreds of millions of years of evolutionary pressure to be exactly as unpleasant as necessary to make the organism move, stop, withdraw, protect, rest, heal. If pain were merely uncomfortable, if it were something you could easily set aside and ignore, it would fail at its job. The unpleasantness is not a design flaw. The unpleasantness is the mechanism. It is what makes pain effective. An alarm that you could comfortably sleep through would not be an alarm at all. When you step on something sharp and lift your foot immediately, pain is doing exactly what it was built to do. When a broken bone makes bearing weight agonizing, enforcing the rest that allows healing to occur, pain is functioning as designed. When inflammation makes a joint exquisitely sensitive to touch in the days following an injury, ensuring that the area is protected while vulnerable, pain is expressing its protective logic. The behavioral changes pain drives: withdrawal from the source of damage, immobilization of injured structures, avoidance of situations that caused harm, the altered postures and modified movements that protect recovering tissue, are all consequences of a system executing its purpose with considerable sophistication. Pain also drives social behavior in ways that extend its protective function beyond the individual. The visible expression of pain, the grimace, the guarded posture, the vocalizations, signals to others that help is needed. Social support, in the context of injury or illness, has real biological effects on recovery. The communication of pain is not simply a personal response to a personal experience. It is a social signal with survival value, evolved in the context of a species that has always lived and healed in groups. Beyond the immediate biological response, pain also teaches. It encodes memory. It shapes future behavior through the powerful learning signal it provides. A child who burns their hand on a hot surface learns something that no verbal instruction could fully convey, and learns it in a way that is retained, bodily, for a lifetime. The aversion that pain creates is one of the most effective learning tools in the biological repertoire. Cultures and behaviors that protect against harm are, in part, built on the accumulated lessons of pain transmitted across generations. But what makes pain genuinely remarkable, and what modern science has worked so hard to articulate, is that it operates across more than just the biological dimension. Pain is not only a physical event. It is simultaneously a physical, psychological, and social experience, and all three dimensions are real, all three are biological, and all three require attention if pain is to be understood and addressed fully. This is the biopsychosocial model of pain, a framework developed over decades of research that recognizes the body, the mind, and the social environment as equally legitimate contributors to the pain experience. Not competing explanations, not a hierarchy in which the physical is real and the psychological is less so, but an integrated understanding in which all three dimensions interact continuously, each shaping and being shaped by the others. The biology, the state of the tissues, the nerves, the nociceptors, the spinal cord pathways, the brain's processing networks, the hormones, the immune system, the gut, the genetic inheritance. The psychology, the beliefs about pain, the emotional state, the history of trauma, the expectations and fears, the coping strategies, the sense of control, the meaning assigned to the experience. The social, the quality of relationships and support, the responsiveness of healthcare providers, the cultural context in which pain is expressed and received, the socioeconomic conditions that determine access to care, rest, and safety. Remove any one of these dimensions and the picture is incomplete. Treat only one and the results will be limited. Understand all three together and something genuinely comprehensive becomes possible. This is where the science of pain arrives, after all of its extraordinary detail, not at a single answer but at a fuller, more human picture of what it means to hurt, and what it means to heal. And now, at the end of this long journey through the biology and the experience of pain, there is something important to say. Something that follows directly from everything we have explored, and that carries genuine practical weight. Understanding pain changes it. This is not a figure of speech. It is a finding, replicated across multiple research contexts, with measurable clinical outcomes. People who understand how pain works, who know that pain is an output of the brain rather than a direct report of tissue damage, who understand the role of sensitization in context and emotion and expectation, consistently report lower pain levels, use less pain medication, and recover more successfully from both acute and chronic pain conditions than people who do not have this understanding. There is a field called pain neuroscience education, a structured approach to teaching people the science of pain in accessible terms, that has been tested in clinical trials and found to produce significant improvements in pain, disability, and quality of life across multiple chronic pain conditions. Not as a replacement for other treatments, but as a component that makes everything else work better. Why? Because when pain is understood, it is less threatening. And pain that is less threatening is pain that the brain does not need to amplify. When you know that the signal your brain is generating does not necessarily mean damage is occurring, the alarm loses some of its urgency. When you understand that sensitization explains why movement hurts even though the tissue is healed, you are less afraid of movement. And less fear means a less activated amygdala. A less activated amygdala means a lower baseline of threat-driven pain amplification. The understanding itself becomes a biological intervention. Patients who are given clear, accurate, non-catastrophizing information about their pain before a medical procedure consistently experience less pain during and after it. Patients with chronic low back pain who understand the neuroscience of their condition consistently improve more than those treated with physical interventions alone. Education, in the domain of pain, is medicine. One of the most powerful and most accessible truths about pain is this: you can, to a meaningful degree, train your brain. Not in the sense of wishing pain away, or adopting a relentlessly positive attitude, or denying the reality of what is being felt, but in the genuine neurological sense of using the plasticity of the brain, the same plasticity that chronic pain exploits to cause harm, to gradually shift the system in a different direction. Practices that activate the descending pain inhibition system, exercise, meditation, therapeutic movement, genuine social connection, moments of joy and beauty and meaning, are not merely pleasant diversions. They are neurochemical interventions. They release endorphins. They activate the periaqueductal gray. They reduce the baseline activity of the amygdala over time. They rebuild the prefrontal regulation that chronic pain and chronic stress erode. Graded exposure to feared movements, done slowly and carefully, gradually updates the brain's predictive model, teaching it, through accumulated safe experience, that the movement it has been flagging as dangerous is not, in fact, producing damage. The prediction changes. The pain response to that movement decreases. The map is redrawn. This is not quick. It is not linear. It does not work perfectly for everyone or for every condition. But it is real. And it is happening at the level of neurons, synapses, and chemistry. There is also something important to understand about what pain cannot be measured by. Pain cannot be seen on a scan. This is one of the most consequential facts in all of pain medicine, and one of the most frequently misunderstood. Imaging technology, X-rays, MRIs, CT scans, shows structure. It shows the physical architecture of bones and discs and soft tissues. What it does not and cannot show is the state of the central nervous system's sensitivity. It cannot reveal the height of a pain threshold. It cannot display the degree of central sensitization. It cannot visualize the altered cortical map, the neuroinflammation, the reorganized descending pathways. A scan that shows a bulging disc does not tell you how much pain a person is in. Studies have consistently found that the structural findings on spinal imaging correlate poorly with pain levels. Large proportions of people with significant disc bulges, degenerative changes, and other structural findings on MRI report no pain at all. And equally significant proportions of people with severe, disabling pain have scans that appear relatively normal. The person and their scan are not the same thing. The structure visible on the image is not equivalent to the experience being lived inside that body. Pain is real even when nothing shows up. Pain can be mild or absent even when something does. This matters enormously for anyone who has ever been told that their scan shows nothing serious and therefore their pain cannot be that bad. The scan was looking for the wrong thing. The real story was in the nervous system, in the sensitization, the altered thresholds, the reorganized processing, none of which appears on any image currently available. You are allowed to be in pain that cannot be photographed. That invisibility does not make it less real. It simply reflects the fact that the experience of pain lives in a dimension that current imaging has not yet learned to see. So let us bring this full journey gently to its close. We began with a simple, startling idea, that pain is not what most people think it is. That it is not a direct report of damage, not a simple alarm, not a fixed and objective measure of injury. We have followed that idea through the firing of nociceptors and the chemistry of synapses, through spinal reflexes and descending pathways, through cortical maps and glial cells, through the influence of sleep and hormones and gut bacteria and the weather, through phantom limbs and referred pain and the way a lifetime of experience rewrites the system from the inside. And what the science reveals, when you trace it all the way through, is something both humbling and quietly hopeful. Pain is one of the most sophisticated things the human brain does. It is not a failure or a flaw. It is an act of protection, complex, personalized, and deeply shaped by everything that makes each person who they are. Understanding it does not make it disappear, but it changes the relationship to it. It replaces fear with knowledge, and mystery with mechanism. It gives back a sense of agency where pain so often creates helplessness. Pain is real. Its biology is real. Its suffering is real. And it deserves everything that careful science and genuine compassion can offer. But it is not always what you think it is. And understanding it, truly understanding it, gives you power over it.